Thank you, thank you, thank you. It is certainly a pleasure to be here. Get this cranked up here. I'm really excited to have the opportunity to talk tonight to y'all kind of about sort of a topic near and dear to my heart. I'm all excited to talk about what causes autism and how do we get from a very complex set of behaviors, whole set of challenges in social communication and behavior that we call autism. How do we get from genes to that complex set of behaviors? So what I'm going to do tonight is first just say that I've got no conflict of interest to speak to. I've got nothing to disclose. All my funding comes from NIH, Autism Speaks, and the Simons Foundation, as well as a few pennies from the sale of my book. And so that's where my money comes from. I just want to specify that, especially when I talk about the causes of autism. I think it becomes really relevant in terms of where my funding's coming from. So what I'm going to do tonight is really kind of talk about a bunch of different things. I want to start just by giving like a 2-second mile-high overview of what autism is and what I'm talking about when I say autism. Just kind of make sure we're all on the same page. And then I want to talk a little bit about why do I even care? Why are we even searching for the causes of autism? Might we not just want to focus all our energy on treatment or intervention? Just want to talk a little about why we're searching for the cause. I'm going to tell you a little bit about why we know that genetics plays a role in the causes of autism. And then kind of want to talk a little bit historically about what the search for the genes and the genetic contribution to autism has been like, certainly since I've been involved. And then we'll switch gears to go from what we know about the genetics of autism to what we know about the neuroscience of autism, and that's how we're going to link the connection between genetics to behaviors we see. So that's the plan. And if I'm not talking loud enough, just please give a shout out, or if you have a question at any time, please shout out the question as well. I'm happy to answer questions as we move along.
I have a question.
Yes.
Can we get a copy of this PowerPoint sent to us?
Okay.
Yes.
Can I say that we're videotaping this and it will be posted on the website in it takes about 4 weeks to do it. So that will include what's up here. Yeah.
That's awesome. That was great. Question, slide number 1. Love it. So we can keep on going. That's perfect. That's great. Definitely happy to any questions. Great. So when I'm talking about autism, what am I talking about? And I have the term up here, ASD, Autism Spectrum Disorders. For the rest of the night, whenever I say autism, what I'm really talking about is autism spectrum disorders. So currently, when we're talking about autism spectrum disorders, we're talking about 3 diagnostic subtypes. It's autistic disorder, Asperger's syndrome, and pervasive developmental disorder not otherwise specified, or PDD-NOS. Those are the 3 diagnoses that fall into the autism spectrum disorder umbrella. That's changing in just a few months, and we're moving away from all these sub-diagnoses, and I'm happy to chat about why we're doing that and why there's that movement away from that. But we're moving away from that to really talk about just autism spectrum disorder. That's going to be the new diagnostic term that's going to represent all 3 of these currently existing diagnoses. I just say autism for short, a little quicker and easier. So tonight when I'm talking about autism, I'm talking about the autism spectrum, ASD. And what we know is that autism is a neurobiological disorder. It's a disorder that's based in the brain, that's characterized by social communication impairments and the presence of repetitive interests or behaviors. So when we're talking about social communication impairments, things we're talking about are things like challenges in nonverbal behavior, reduced eye contact, challenges in using facial expression to augment communications, reduced or atypical use of gestures to communicate. We're also talking about in that social communication domain. We're talking about things like challenges with reciprocal conversation, challenges with establishing and maintaining friendships, that sort of back and forth social engagement, so social communication. In that second domain, the domain of restricted repetitive behaviors and interests, we're talking about things like interests that are unusual in their intensity. So for example, a youngster I used to work with who was 13 years of age, knew everything about cameras you could possibly know about cameras, everything, and he wanted to talk about the different types of lenses and the switches and all the buttons, and I don't even know, and I couldn't even follow along. He knew everything he knew about cameras. Very typical hobby to know about cameras, but he took it to the nth degree. Conversely, you might have interests that are unusual just in their focus. So another child I used to work with was very interested in toilets, and he just loved to look at toilets and talk about them and find out what kind of make they were, which I didn't even know that you can actually find that on toilets, but there's a little label right there. So anyway, interests that are unusual in their focus. Also in this domain, we talk about things like repetitive motor mannerisms, hand flapping or finger flicking, some terms sometimes used, challenges with changes to routines, disruptions when there's a transition, as well as unusual sensory interests, either aversions or sensitivities. So those are things we're talking about in these two different domains. Also, what do we know about autism? Again, this is the mile-high overview of what autism is. It's a disorder that affects boys more often than girls, 3 to 4 to 1. We know autism is found in all social classes and all racial ethnic groups, does not discriminate. We do know that behaviorally based interventions are effective at improving outcomes in autism. And most interventions seem to be associated with improvements for many individuals, but no interventions at this point seem to be appropriate for all kids with autism. So speaking to the heterogeneity, the variability in autism, I'll talk a little bit more about that. Currently, there's no genetic or biological assay to diagnose autism. There's no genetic test. There's no imaging test we can do, no blood test. The diagnosis is simply made based on expert observation of behaviors in these two different domains I talked about, social communication and repetitive interest, or restricted interest behavioral domains. Lastly, we know there's a strong genetic component to autism. That's the kind of mile-high overview. So I want to jump in and dig into the causes of autism and what we know causes autism. But first I want to talk about why we bother searching for the cause. So right now, on average, around the country, the average age at which a child is diagnosed with autism is between 3 and 4 years of age. That's kind of where we're at now. We've come a long way in the 15, 20 years that I've been doing this sort of work. Back then, average age of diagnosis was well into middle childhood. There are certain places we're doing much better than that. Here in Seattle, we can, yes! That was my question. Yes, so certain places we're able to diagnose and have an average age of diagnosis much earlier than that, 3 to 4. That's kind of when you look at the country in total.
Do you know what the average is?
In Seattle? I don't know, and I would love to. It'd be sort of tricky to sort of touch base with all the different centers and folks that are doing diagnosis here and sort of try to get a sense of that. It'd be a great question. Certainly find outable, just don't know. So that's where we're at right now. We know on average that parents first start noticing that something's not quite right with their child's development at 17 months of age. So we're looking at kind of a gap there when parents are saying to themselves and to their pediatrician, gosh, Jimmy's just, I think there's something not quite right. He's not using language the same way that I would expect my other child does. Go. It takes a little while from that 17-month age point when parents first notice something's not quite right till this diagnosis at 3.5 years of age. So the problem is then, at 17 months of age, we're noticing atypical behavior, that is delays in language, is the number one sort of referral reason for parents currently. By the time parents are noticing this, and certainly then by the time we make a diagnosis, developmental trajectory is kind of veered off out of the norm, right? In order for us to notice that something is delayed, we've already gotten off track. So we end up not detecting and diagnosing autism until child's development is well out of the range of normal. And then, of course, we intervene at that point. And that's great, we're intervening, but we're still not intervening until 3 years of age. So by identifying the potential causes of autism early on, if there's a genetic contribution, which we know there is, if we can identify that earlier on, we can detect and diagnose autism really at birth, and ideally then intervene right away, and maybe even in fact prevent the development of autism. So again, the idea there with early detection, we can intervene earlier, and ideally, in theory, we'd have a better outcome. I think a good example of this is PKU. So many years ago, PKU, kiddos who had PKU would actually go on and they would actually get oftentimes a diagnosis of autism as well as intellectual disability. So I think it's a really perfect example in this case. But at birth now, every kiddo gets a little heel stick. And we can check and find out, does this child have an inability to process phenylalanine when exposed in his or her diet? If so, we can completely eliminate that phenylalanine for that child's diet, and we don't see any of the phenotype. That is, we don't see any of the intellectual disability and autism symptoms that we see. So it's a really great example of if we can do the same sort of thing with autism, we can maybe really help out quite a bit. So that's one reason why I want to be focusing on the cause of autism. We can help with early detections, we can intervene earlier. The second is it might help us understand the mechanisms, and that's certainly kind of the work that I'm most interested in, is helping understand the mechanism of how we get from a disrupted gene or some sort of genetic variation to behavior. By understanding what genes are involved in autism, well, that helps us understand what proteins they code for, the molecular pathway, and therefore then the mechanism for behavior. Throughout all of this, then, that can help us figure out what some better pharmacological interventions might be, intervening at a variety of different levels there, either at the genetic level or at the molecular level. So again, that's kind of the basics for why we're doing this, as opposed to jumping straight into intervention. We know that interventions do work, and so we want to focus on enhancing which interventions are working for certain kids with autism, but I think this is one way to do that. So that's kind of why we're doing this. But how do we know genes are involved? Well, the story there really starts back in the 1800s. And bear with me as I sort of go through this little saga. So, and the reason I say it starts in the late 1800s is because I want to provide a little context for what happened in the 40s and 50s and 60s and 70s. So really back in the late 1800s, Freud's sort of notion that childhood disorders are really a product of essentially the parent environment, right? So this was kind of the thinking in the 1800s that really held sway up until the 40s and 50s, that really any childhood disorder, atypical behavior seen in the child, because of that sort of that cultural understanding about childhood disorders, really a lot of the sort of blame, so to speak, went to the parents. So I want to provide that little context for when I kind of tell the story a little bit. We first hear the word autism back in 1911 when Eugene Bleuler, who was using the word autism really specifically in reference to individuals with schizophrenia, talking about the inward turning of the self. But that's where we first hear the term in the scientific literature. Go, a few years up to 1943 is the first publication by Leo Kanner regarding individuals with autism. 10 years later, double helix is discovered. We start to understand a little more about genetics that's happening. But really what was going on between 1943 and 1967 were folks saying, gosh, so Leo Kanner's been talking about autism. There's a number of individuals, they have impairments in social communication, the presence of these restricted interests and behaviors. What's going on? What might cause this pattern of behaviors? Again, because of that cultural zeitgeist wrapped up in Freud's sort of notion that all childhood disorders result from childhood and the parent-child relationship, folks started looking to the parents. And Bruno Bettelheim kind of spearheaded this idea. Bruno Bettelheim was a professor in Chicago and who coined this term, refrigerator mothers. And his idea was that what was happening in autism was that kids with autism, when they were young and they were sad and they were reaching out to parents, that moms, specifically moms and dads, but really moms, that moms' cold responses to them forced these children to, when they were sad, to turn inward because they weren't being comforted by moms and dads. And he published this book, The Empty Fortress, kind of highlighting this notion. And what I think is sort of interesting about Bruno Bettelheim, he got a lot of sway, got a lot of attention for this book and his theories. But it's interesting. He came from Europe, and he had we sort of start to look back in his history, and it turns out he said, "Hey, I've got this Ph.D. in psychology. I'm a professor. I'm going to run this whole clinic here for kids with disabilities." And you start to look back into his history and realize, like, gosh, wow, this guy never went to school. He actually doesn't have a Ph.D. Wow, how did this guy get this position of authority running this school for kids with disabilities? And really, he's just making stuff up left and right. So it's sort of interesting when you sort of start to dig into the history of Bruno Bettelheim. But anyway, his theory held sway for a long time. And it was really rather unfortunate, as we imagine, as parents are struggling and feeling so so guilty and horrible about sort of being accused of being cold and essentially unwarm to their children. So that was going on in the 60s. There was, but not everyone sort of held on to Bruno Bettelheim's belief. Certainly a lot of folks in the field did, but not everyone. And there was one young psychiatrist who thought that this actually was total malarkey, and she said, okay, I think there's a real strong biological component to what's going on with autism. And so she finished medical school. She headed over to England to work with Michael Rutter, who has since been knighted. How awesome is that? I want to be knighted for doing science. How cool is that? So anyway, Sir Michael Rutter. So she and Sir Michael Rutter decided to use the cutting edge genetics technology at the time, which was a twin study. And so what Susan and Michael did is they rounded up all the individuals that they could find, all the twins that they could find, where at least one twin had autism in England. And she went around, driving around England as a resident and fellow in psychiatry, and interviewed all these families to find out, okay, make the diagnosis and meet with them and include them in this study. And so let me tell you a little about what we found, or what she found in that initial study. So what she was comparing, again, this is the technology of the day in the 70s, which is cutting-edge genetics technology. She simply looked at concordance rates for monozygotic twins, those are identical twins, compared to dizygotic or fraternal twins. And she just simply counted up, well, how many times when I find one twin with autism, how many times does that other twin have autism? And so what she found was that 60% of the identical twins or monozygotic twins that share almost 100% of their DNA, not exactly, but they share a lot. 60% of those twins both had autism, compared to the dizygotic twins or fraternal twins, which share about 50% of their DNA, just like any other siblings. That rate was less than 10% for the dizygotic twins. So in 1977, boom, we get this indication that something's going on, because identical twins seem to have a much higher concordance rate for autism. Nowadays we could think, well, sure, yeah, that's whatever, fine, that's cool. But back in the 70s, this was sort of mind-blowing, because so long people were thinking this is caused because parents were just being cold. But this study was able to sort of put the nail in that coffin and say, you know what, we're barking up the wrong tree, folks. Sorry, Freud was great and all, but he's kind of missed the boat. And really what we're talking about is a biological disorder. So Tony Bailey, also in England, hasn't been knighted, but also in England doing science. He followed up with this sample over the next 25 years and continued working with families to sort of fill out the sample, increase the sample number. But in that time that Tony Bailey was doing this study, we had some changes in the way we diagnosed autism. So back in the 70s, we just had essentially autistic disorder as the diagnosis. Subsequent to that time, in the DSM, we added these diagnoses of Asperger's syndrome and pervasive developmental disorder not otherwise specified, as I mentioned. And the criteria for the diagnostic criteria loosened in that time. So when you include not only individuals with autism in that twin comparison, as Tony Bailey did, he found that the rates shot up even higher for the identical twins and really didn't change much for the fraternal twins. And then also, what Tony did was he said, hey, you know what, I'm also just going to include in this comparison individuals where one of the siblings doesn't quite have autism but has other traits that are related to autism. So might be uncomfortable in social situations, might have increased anxiety, might have some troubles with transitions, but doesn't quite meet full criteria for autism. And he termed this, as many folks did, called the broader autism phenotype. So it's symptoms and qualities and characteristics that are similar to autism but don't quite meet that full threshold for a diagnosis. When you include individuals like that in that sample, the concordance rates bump up to 90% for the identical twins and still kind of hover around 20% for the fraternal twins. So with just some very basic twin studies, we're able to highly demonstrate what's going on in terms of the genetic contribution to autism. So just last year, or I guess 2 years ago now, Joachim Hallmeyer did a new twin study, a population-based twin study, where he worked with every possible twin family in the state of California where at least one twin had autism, and it did the exact same thing that Susan Folstein did. Just looked simply at concordance rates from monozygotic or identical twins and fraternal or dizygotic twins. This study got a lot of attention in the media, I think kind of in not the best way, mainly because they found very similar rates in terms of identical twins and in terms of the autism and autism spectrum. But they found a much higher rate of dizygotic twins than had previously been talked about. So about 35%, 33, 35% of these fraternal twins both seem to have autism. So much higher than the original projections of somewhere between 10 and 20%. The media jumped onto this that year and said, holy cow, all you scientists are missing the boat. Autism is a disorder about the environment, because look at these high rates for these dizygotic twins. It's crazy, oh my word, look, it's so different now. They missed the boat still, because really we're still seeing a pretty significant contribution there in monozygotic twins. So I was a little disappointed the media kind of jumped onto that notion. I think it's important for us to talk about environmental contributions. I'm going to talk about those today. Certainly genes don't act alone, they always act with their environment. But I think the media got hold of and started talking about different types of environmental contributions as opposed to things that I think we should really be thinking about. So all this is to say, we now know, we knew in the late 70s that there was certainly a genetic component to autism. There was one other way we sort of more recently we've been thinking about, we know that genetics are playing a causal role or contribute a role in autism, and that's from Sibling Studies. So there's been a real movement in the past 5 years, 7 years, to look at and study infant siblings of children with autism. So that is, family goes on, a child is diagnosed with autism. Because of these rates that we talked about here, these higher rates in the twins. Oh yeah, look, there's a little pointer, perfect. Because these higher rates that we're seeing here, we know that there's going to be a greater chance that a younger sibling of a child with autism is going to develop go on to develop autism, and so we can study these infant siblings. And from these infant sibling studies, we've learned a lot. We know that if a family has one child with autism and then has a second child, we know that the recurrence risk, the risk for that second child to go on to develop autism is somewhere between 10 and 20% depending on the study. We know that if a family has two children with autism and then goes on to have a third child with autism, that recurrence risk bumps up to 35%, which really kind of mirrors what we're seeing in that twin study that Joachim Hallmeyer reported. So these two very behavioral ways of looking at genetics, we can now see, wow, genes are playing a strong contributory role in autism. Twin studies and these sibling studies. So we know they're involved, but what do we actually know then about the genetics of autism? What do we know about the nitty-gritty of it? And so I thought I would tell that story kind of in a little timeline again. And we'll start the story not in 1977, because I was only 5 then. I was playing kickball, if that. So I was really not thinking about autism at the time. But my story sort of starts in the mid-90s, which is when I got involved in autism genetics, which, interestingly enough, was really when things took off with genetics. I'm not sure, it might just be a correlation, I don't know. But I just want to highlight that things really took off with genetics of autism in the mid-90s. Has nothing to do with technology, I'm sure. At any rate, so in the mid-90s, I linked up with Susan Folstein, and we worked on the collaborative linkage study of autism. And so at the time, we were using kind of the best cutting-edge technology in genetics, which was to round up multiplex families. That is, families where there is at least 2 individuals with a diagnosis in the family. So it could be 2 siblings, could be a dad and a child. You just need to have 2 individuals in the nuclear family to do this linkage analysis. And what linkage analysis essentially is what we would do is we would gather up these families, again, multiplex families where there's 2 individuals who have been diagnosed with autism. And I'll spare you all the crazy mathematical details, but essentially what we're doing is we're looking for markers in the genome that both individuals with autism share that aren't shared by the other individuals that are not affected in the family. And by identifying which markers are shared, we can figure out essentially where there is likely that there's a gene or genes playing a contributory role to autism. So the linkage analysis essentially says, it kind of points us in the neighborhood, like whereabouts in this part of town are we looking for the genes that might be involved in autism. So again, it's not going to pinpoint genes necessarily. It could if you had a massive sample, but essentially what we were doing is it would point us in the right direction. Maybe identify particular region of a particular chromosome. Then we'd look further about what genes are involved and localized to that region. That was kind of cutting edge at the time. The idea what was going on, the thinking in the time of the 90s, what was going on in terms of the genetics of autism, was that really we're looking at a model where multiple genes were acting in concert. And essentially, if you got the right recipe, you got the right concoction of genes from mom and dad, that would result in autism. So here would be the example. If we think back to sophomore year in biology, so sort of take yourself back. For me, I have to dodge Mr. Messenger's spittle because he would spit a lot and I sat in front of the class. It was always rough. So I try not to take myself back too often to biology sophomore year. Anyway, take yourself back there. Your biology teacher's talking about Gregor Mendel's peas, right? So we got these peas. Each of these peas has a gene for color. So we could say a gene would be marked as gene locus A here, in this case, on this little sample here. And there's going to be variations of those color, of pea color, right? There's going to be a yellow pea or a green pea. And so we could think about those are the alleles, the variations. We think about that as these two different versions of this gene A. So in this case, there's a big capital A and a lowercase a. So the theory about autism was that, again, this is just using 5 genes as an example, was that if you got a certain collection of autism risk genes, that is autism risk alleles, variations at a certain gene locus from dad and a certain combination from mom, that would result in autism. So in this case, in this little example here, because each individual is going to get an allele from dad and allele from mom. In this case, this first child happened to get, he had a 50% chance, he happened to get the capital A from dad and the lowercase from mom, capital B from, and lowercase b from mom, capital C from dad, lowercase c from mom. In this case, because there's only lowercase d, you got got the lowercase d and e from dad and uppercase D and E from mom. It just so happens because he just, by random chance, he got this combination. He ended up with autism. Same with his sibling. And yet he has a third brother who only got the capital C, capital D from mom. Does that make sense for sort of thinking back into sophomore year biology? So this was the thinking. Multiple genes acting together. If you got the right allelic variation of a certain gene, that would confer autism risk. It didn't mean these genes were broken. It means that the genes were working just fine. It just so happens that you had the certain variant that increased your risk for autism. That was the thinking, and that's what we were going by with our linkage study approach. So what we found back, and we published back in the late 90s, was that there are 2 regions for which there was, we thought, increased likelihood that there was genes localized to that part of the genome. So I won't spend too much time, but I'm going to just briefly kind of show you what this is here. These are just little figures showing 2 different chromosomes. This is chromosome 13, chromosome 7. The x-axis here would be the length of the chromosome, and it's marked by centimorgans, which is the distance between markers. And then the y-axis is the likelihood that there was something going on in that particular region in a gene. The take-home was we found in our sample, we found high likelihood there was something going on in chromosome 13 and high likelihood there was something going on in chromosome 7. What was exciting about the chromosome 7 finding were 2 things. One is that there was another group in Europe that found a very similar thing in the same year. And also chromosome 7 has been linked to language impairment. So we got pretty excited about this finding and kind of people all got on board. What got then tricky about that is in the time subsequent to that, lots of different people were publishing their different linkage studies, and no one was finding the same thing. So no one was able to find what we found. We weren't able to find what anyone else was finding, and no one found out anything that was the same. It was kind of a nightmare with linkage analysis. So essentially we had each new study where everyone was doing great work. You know, Yale was doing great work, UCLA was doing great work, UW was doing great work, we were doing great work in Boston. We were all doing great work, but with samples that are in the order of 100, 200 individuals, and we couldn't seem to replicate any findings. So that was going on for many years. Good 6, 7 years, folks were publishing the linkage study analyses and were saying, gosh, wow, we're getting close, we're finding neighborhoods, but we can't seem to replicate. It seems unlikely that this is going to account for everything that's going on in autism if no one can replicate the findings. And then things took a drastic shift in 2007. So Susan Folstein, I'm going to take us back to the 90s again. I'm there working with Susan Folstein. I'm driving all over New England, drawing blood from kids and back seats of cars and the kitchen table on the floors. Work with families on making these diagnoses. Work with hundreds of families throughout New England. We're working on our collaborative linkage study of autism. And Susan says to me, she says, Rafe, this is great that we're doing linkage analysis. It's great that we're focusing on multiplex families. But multiplex families only account for about 20% of all cases of autism, right? The vast majority of families don't have more than one child with autism. They have one child with autism. So I wonder if there's something going on there. Were we missing the boat as a scientific field if we're only focusing on this 20%? So she said, Rafe, here's an idea. I want you to drive all over New England again, but this time only work with families where there's one kid with autism, and then see if you can find families where there's a sibling who's kind of like the opposite of autism. So try to track down kids who are super social and super flexible, easygoing, hyperverbal, in sort of a social communicative way, and find these folks, and then let's work with these families. So go, I did that, spent a bunch of time driving around New England doing that, and it was great. Worked with a lot of families. It was super fun, just like the previous study. And then I took a little hiatus from research and went and did Peace Corps. And so I kind of left the autism world behind for a little while. And in that time, Jonathan Sabat, who, along with Michael Wigler, who's kind of like this rogue geneticist from Cold Spring Harbor back east, said, hey, Susan, I know that you and Rafe drove all around New England and got a whole bunch of families. He said, hey, Susan, I know you, Rafe, but really, she didn't drive anywhere. But at any rate, so, hey, I know you guys collected all these families. Hey, I think this might be, we've got some new technology available. Let's look at these families that you've studied. The new technology that was available was looking at structural changes in the chromosome. So it's a very different way of thinking about the genes that are involved in autism. We're no longer looking at variations of genes like in this type of study. We're now looking at structural changes to the chromosome themselves. And we knew back in the 80s, we started doing this back with electron microscopes. We could look under microscopes and we could actually see structural changes, duplications, deletions, events where there's like a chunk of DNA that's missing or a chunk that's been duplicated. So for a long time we could do that with microscopes, but you could only see that, things could only be that small before you wouldn't be able to see them anymore. So with some new technology, we were able to identify these structural changes that are much, much tinier and smaller. So what Jonathan and Michael did was they took our sample of individuals, these multiplex families where there's 2 or more individuals diagnosed with autism, and then looked at this other sample of individuals who worked with these simplex families where only one child was diagnosed with autism, and just looked at the rates of these copy number variations. And this is just sort of a picture to show you what I mean when I'm talking about copy number variation. So I'm talking about breaks either that lead to a deletion where there's a chunk of DNA that's missing. These are little cartoons of chromosomes here. And when we get a little deletion, this little chunk disappears, and we essentially get a shorter chromosome. And with duplication, we get that break point is then duplicated, so we get additional information essentially on our chromosome. So there's a lot of different types of structural changes we could have. These are kind of the 2 most common: deletions, duplications. This is just to give an example of one of the more common events that we find. So here we've got our cartoon of a chromosome, and just to kind of jump back, if we think about it, there's 23 pairs of chromosomes. Again, we're going back to sophomore year biology here again. 22 autosomes, those are all numbered 1 to 22 in order of size. 1's the largest, 22 is the smallest, and then a pair of sex chromosomes, X, Y. So here we've got an example of chromosome 16, so the 16th chromosome. All the way chromosomes they're set up, there's a short arm and a long arm. They're joined together at centromere here. The short arm is labeled with the letter P, stands for petite, and Q stands for, doesn't really stand for anything, but it's the long side, the long arm of the chromosome. So when we talk about regions of chromosomes, we're talking about chromosome 16, P11.2. What that means is we're talking about the 16th chromosome, the short arm of the chromosome, and this neighborhood, the band is 11.2, and this simply refers to if you stain a chromosome, look at it on a microscope, there's a certain number of bands, and so we're just labeling each of these bands. So at this particular region, 16p1.2, we see deletions and duplications, and this is just a pictorial representation of what that deletion readout looks like. So we have a chunk of this chromosome is missing for this particular individual here, highlighted in red. These are all the genes that are localized. There's 29 genes localized to this region. So Jonathan Sabat, Mike Wigler, take the sample of multiplex families, sample of simplex families, and they looked for these deletions and duplications in these different families. And what they found was that in our multiplex families that we've been studying for 10, 15 years at this point, they found that the rate of these mutations, these deletions and duplications that are arising de novo, that is, mom and dad didn't have these genetic events, the child with autism had these, found a rate of about 3% of these deletions and duplications. In the simplex families, that rate jumped up to 10%, as 1 in 10 of these individuals with autism had a de novo mutation that involved a structural rearrangement to the chromosome. And just as a reference point, typical individuals have these copy number variants. They're called CNVs, these deletion duplications. About 1% of all typical individuals have a CNV like of this size. Really what CNVs are all about is where they're located, right? So you could have a CNV that is the same exact size as, say, an individual with schizophrenia or ADHD or autism, all disorders that have been tied to copy number variants. You could have a CNV the same size, but if it happens to be in a chunk of your DNA where there's no genes involved, well, we don't really know what that means, but the decreases likelihood, though, that you will have a disorder if it's in a chunk of your DNA where there's no genes localized to that region. So that was 2007. So that really ushered in a whole new way of thinking about autism research and genetics of autism. Prior to that, again, we've been doing linkage analyses. We've been focusing only on multiplex families. And now these sort of rogue geneticists said, hey, we really got to focus on the 80% of individuals we haven't been thinking about for the past 20 years. So the other thing that was interesting about this study was that the senior author, Michael Wigler, this rogue geneticist, happened to have a good buddy who had a daughter with autism. And this good buddy happened to be the 49th wealthiest person in the world. And this good buddy happened to decide he wanted to take his foundation that was focused on science and math and direct all those funds to autism research. So what Jim Simons did is he pulled together, because he has a boatload of money, he pulled together all these Nobel Prize winners from all different fields of science, neuroscience, genetics, all over. And he pulled them into a room in New York City, and then he had two people representing the autism community. One person was Jerry Dawson from here, and the other person was Ami Klin, who used to be at Yale and is now at the Marcus Center down at Emory, and said, gathered everyone in a room and said, okay, Jerry and Ami, you guys tell all these world-class Nobel Prize winners what's going on with autism, and then these world-class Nobel Prize winners are going to tell us what we need to do. A kind of interesting model, a little bit different, a little bit goofy, but at any rate, brought that together, and ultimately what came out of that think tank meeting was the take-home was that we really need to understand the causes of autism so we can actually figure out how to intervene most appropriately. So Jim Simons says, okay, we're going to do something about this. We've got to figure out the causes of autism. He turns to his good buddy Michael Wigler with kind of his crazy hair and his sort of like, kind of crazy lifestyle out there on Long Island as a geneticist, and he says, okay, great, I'll tell you what we need. We need 2,000 families. Oh, scratch that. 3,000 families. He changed his mind halfway through. He said, we need 3,000 families where only one child in the family has autism. And we need these families really fast. And so what we'll do once we get all these families, we need blood and we need really good, clear characterization of all the characteristics of these families. And then we'll run these cool analyses, the CNV analyses, and see if we can figure out what's going on with autism. So Jim says, okay, great idea. What should we do? He decided that, well, Yale was doing great work. They've been doing this great linkage study. UW, we were doing this great linkage study. UCLA was doing some great science. But we're all doing great science on our own. And so Jim Simons says, all you crazy scientists, can you guys work together? We got a big issue here. We got to figure out what's going on with autism. How about you all work together? And I'll make sure that the funds are there so you guys can actually all work together and work together well. And so he pulled together the 12 sort of top research institutions that are focused on autism in North America and got everyone working together for the first time. And we rounded up a whole bunch of families, again, close to 3,000 families, where there was just one individual autism. And we, thanks to everyone's grateful time and donation, these parents are willing to come on in over and over and give us their blood and give us their time. We could do all this testing and actually collect, for the first time, a very large sample where everyone in all the different centers were doing exactly the same thing. And we made sure we were all working together because we would send videotapes back and forth of us doing these evaluations to make sure that no one site was doing goofy things. We're all doing everything exactly the same way. So it's a real change, a real shift in the way autism research is being done in general, and has really been a great opportunity for us to actually advance the field significantly. So actually, in a couple weeks, we'll be publishing our paper where we analyzed all the CNVs, again, these duplications and deletions in this whole big sample of 3,000 individuals with autism. This is just kind of a busy little figure, a busy slide to highlight what we found. So when we scanned through, it turned out to be just under 3,000, about 2,600 individuals. We said, great, can we identify any recurrent copy number variants that seem to be happening in this sample of individuals with autism? And so in order to make better sense of that story, we would then also compare it to a very large sample of individuals with intellectual disability and then a whole bunch of control individuals who don't have any psychiatric disorders. Really the take home of the study is there are a number of copy number variations that seem to be accounting for autism. Again, we found very similar rates to that study that we found originally back with that sample that I collected in New England of about 10%. That is, about 10% of these simplex families seem to have a structural change to the chromosomes, to their chromosome, that results in ostensibly autism. So that's a little preview that should be coming out in a couple weeks. But I want to jump back from that and talk a little bit more about some of the other stuff that I think is actually a little bit cooler than that finding. So I'm going to take us back then to the time intervening where we're just getting the Simon Simplex collection study started. So back into 2009. So while these 12 different centers were getting started, we're doing our genetic studies, we're working with families, we're getting started all working together. Another group of us decided we would kind of pool our resources and pool all the information that we'd gathered together, even though everything wasn't collected exactly the same way, and so it's kind of hard to compare folks, kind of like putting apples and oranges and pears and grapes all in the same bucket instead of apples and apples like the Simon Simplex collection. But we thought at least this is a start. And so we all put our apples and oranges and apples and pears together, that is families that participate in our studies at our different sites. And we pooled all that information and did what's called a genome-wide association study. And what a genome-wide association study is essentially you find all the individuals in a very large sample. In this case, we had 6,000 individuals with autism. And we scan through everywhere in the genome, and we just try to identify what points in that genome, by looking at particular markers, what points in that genome might there be a gene or genes that are playing a role in autism. So in this very large sample, it was the largest sample at the time that had ever been published, largest collaboration. What we found there's a particular region on chromosome 5. Well, actually, we can all just know what this means now. 5p14.1 just means the small arm of chromosome 5 at in the neighborhood of banding of 14.1. At any rate, in that region, we found a likelihood that there's something going on there. That doesn't sound all that different from all the linkage studies we were doing back in the early 2000s, but what was different about this study was, well, first it was very large, so we were actually able to make sense of the data. The second was we identified that there were two different genes localized to this region, two different cadherin genes. And cadherin genes are involved in the way neurons connect and communicate with each other, the way they join forces. And so for the first time, we then had a real clear potential biological mechanism for what was going on in the brain. Now we found, so it seems like in this very large sample, we've got these genes that are involved in the way brains are connecting and communicating. And so we said, maybe that's really what's going on. It really has to do with the way brain cells are communicating and connecting and joining their synapses. So it caused quite a big stir because that we had, for the first time, a potential mechanism for what was going on in autism. Time magazine said, hey, this is a top 10 study of the year. It kind of got a lot of excitement from the media, which I was excited about because the media was excited about something good and exciting, not just following Jenny McCarthy's ideas, which are fine, but just different from science. At any rate, so that was 2009. We got very excited about that. But then we weren't going to stop there because there was some new technology on the rise. And the new technology was our ability to sequence the full exome. And what that is, if you think about our genome, we think about all the different DNA base pairs. Again, we're going back to sophomore year biology, all those different base pairs. There's parts of our genome that we know code for genes. There's parts that code for little amino acids which form together, form proteins. Those are the genes. Those are called exons. There's a whole big bunch of our genome. We don't understand what it does, called introns. It's a whole bunch of base pairs there, excuse me, that don't seem to account for, that don't make any genes. I'm sure there's a reason for this. No one knows exactly yet what all these regions do. But we do know what the exomes do, and that is they code for genes. So with some new technology, we were able to go ahead and fully do exome sequencing in a sample of individuals with autism. So this was really spearheaded by a great colleague of mine, Evan Eichler, who's here at UW. And what we did was simply we just, again, sequenced all the gene coding regions in the genome. So this is just kind of a complicated slide from NIH, which really just highlights we have our base pairs in these little codons, collections of 3 base pairs form together amino acids. You get a string of amino acids, they form a protein. I don't want everyone to fall asleep, so I'm just going to say this really fast. So what you do is you sequence all these regions that are pulling together these amino acids to form proteins, and you find out when there's some sort of mistake, essentially, or a mutation where there's a change in those base pair that results in the truncation of that gene, or it just basically tells it to stop making that protein. Basically, it breaks the gene. Really, the take-home, the real short message is we were looking for broken genes. That's essentially what we're doing. So what we did was we selected out 20 individuals that we thought might be likely to have some sort of new event. And the reason how we picked that out was we looked for families where there was just one child with autism and there were other siblings that didn't have autism. We looked for families where there's no family history of autism anywhere. So it wasn't out there, and it wasn't Uncle Jimmy didn't have autism and it was just undiagnosed. It wasn't Dad who was struggled with social interactions. And really we looked for folks where there was no history of autism anywhere in the family, and it was sort of arose anew. And what we found in that first sample of 20, we went through and scanned the exome, was essentially we found that in 4 of these 20 individuals, we found broken genes. We actually able to identify, wow, in 4 of these 20 folks, 20%, there's something broken. This gene is not working. And we were actually able to identify what these 4 genes were and understand what they did. All 4 of these genes played a role in the way your brain cells communicated with one another. So really kind of highlighting this notion that we're looking at the way brain cells are communicating, and that's kind of what may be playing a role. It also happened that these 4 individuals were more impaired than the other 16 individuals in this sample. This is just to highlight that in terms of full-scale IQ, cognitive ability. Excuse me, the starred individuals, or the individuals with the gene-disrupting mutations, they were more impaired cognitively, and they had increased autism severity, as measured by the Calibrated Severity Scale, the CSS. So this study also got a lot of attention. We published it in Nature, got a lot of attention from the autism world, because again, we were able to identify genes, actual genes, not just regions of the genome, but actual genes that weren't functioning properly in individuals with autism, and provide more insight into the mechanism, again, the way brain cells are communicating with one another. So we got so excited about this with our sample of 20 individuals, we thought, what if we did this with 200 individuals with autism? Granted, that costs millions and millions of dollars, but if we found that money and we did it with 200 individuals, would we be able to answer the questions? Would we be able to actually figure out, wow, what are maybe all the genes that are involved in autism? We got really excited about it. We went through, found the money, sequenced all 209 individuals with autism. We did the exact same study. What we found in the sample of 209 individuals, we found 248 different gene-disrupting mutations in about 25% of the sample. So about 50 of these individuals had a number of neurologically expressed gene-disrupting mutations. We were excited, for sure. We found now this is about 25%. We're able to identify what's sort of atypical in the genetics. But then we thought, well, gosh, what's going on with this other 75% of the sample? We thought we'd figure it all out. We'd identify all these different genes, but really we only found what was going on in 25%. And then we said, let's figure this out mathematically. We have 200 individuals with autism. There's a potential of this many thousand genes that are possible. Based on that, based on the number of mutations we found in our sample, we estimated there's probably about 800 or so different genes that are involved in autism. So we were actually not excited at all. We were actually crushed, to be honest with you, because we thought, gosh, how are we ever going to make sense of this? There's 800 potentially different broken genes that may be playing a role in autism. Wow, kind of mind-blowing for us. It helps me to, though, think about how complicated this whole story is. So there were a couple of things we found out about this that came out of the study that I think are really important. The first thing was this. Wow, look at that complicated web. So exciting. No, seriously. What's exciting about this is, well, I could just say with this. This is a protein network. That is, how do different genes interact with one another? And it turns out that of all those different gene mutations that we found, 44 of those genes were part of this tightly interwoven network. This particular network is called the beta-catenin network, which is involved, again, it's no brainer now, but involved in the way that brain cells communicate and connect with one another. So that was kind of exciting for us to see, wow, okay, there seems to be a real tight network that's playing a role, a causal contributory role in autism. The other piece we found is just that the number of mutations that an individual has increases the challenges or impairment that an individual has in terms of the severity of symptoms, in terms of cognitive impairment. So essentially the take home is, if you have one gene mutation, that's problematic, and it was going to result in a series of challenges. If you have 2 or 3 or 4, it's going to increase the challenges you may have. Again, all makes kind of sense, but we actually had science to back that sort of notion up. So, but we didn't stop here. So we now have a network of genes we want to explore. We've identified are playing a causal role in autism. So then what we did is we decided to say, is there anything even newer? Is there any more cool technology we can use and apply to this? And it turns out there's this a neat technology called the molecular inversion probe, which is a very fancy term. We call it MIP sequencing. But what we can do with MIP sequencing is you can scan through genes of interest in a very large number of individuals very cheaply, essentially. So again, we're talking about exome sequencing. We're talking about $6,000 per individual. So per family, we're talking $25,000 to do the sequencing. With MIP sequencing, you can do it for under $100 for a whole family, and you can sequence a whole family for particular genes of interest. So you can't sequence all the exome. You can sequence the genes of interest. So we thought, why don't we start with the 44 genes that are part of our network and sequence, let's say, 2,000 individuals and see if we can figure out what's going on there. So we did that. This paper just came out, oh, it says 2013, but it was actually late 2012. Anyway, that came out pretty recently in Science. And what we found in the study was, we tested, using this molecular inversion probe, we tested 2,000 families for the presence of gene-disrupting mutations in those 44 genes that are of most interest to us. What we did was we identified the recurrent mutations to these. So these 44 genes, we found a number of these broken genes were broken in a large number of individuals. And when we tallied all those up, 6 of those 44 genes alone, the most recurrent disruptions to CHD8, disruptions to DYRK1A, a couple other genes, that just those 6 genes alone, when we extrapolate out, account for 1% of all cases of autism. So 1% of all cases of autism doesn't sound like a whole lot, but really when we're talking about 800, 900 different genetic mutations that might result in autism, when we talk about 6 specifically that are accounting for 1%, that's a pretty sizable contribution. So we are very excited about that, and we're really thinking that these 6 particular genes, high-frequency genes, are the things we really need to understand better and more. They're going to help us really understand how do we get from genes to behavior in autism really kind of the best way. The other little bits of information I think are sort of interesting about the study was one particular metric that we used, one particular phenotype we used, that is expression of a genotype, was head circumference. Excuse me, head circumference is a good proxy for brain size, actually, and it's something that we can do pretty easily. We can gather those records from physician's office. We can do it very quickly in our own office. And what we found, interestingly enough, were 3 of these top genes were tightly linked to head size. So cadherin-8, which is here highlighted in red, one of our more common recurrent genes, all these individuals had head sizes that were much larger than a typical individual, and much larger even than much larger than individuals with autism, who often have, on average, a larger head size. We found some individuals with a P10 mutation that had even larger heads. All this is to speak to a couple different things, and I'll sort of jump back and forth. As I mentioned, head size is a proxy for brain size, and if we're thinking about the way the brain is communicating and making connections, one might imagine that what might be happening in some individuals is we get brain overgrowth, and that the connections between neurons aren't being effectively pruned and cleared away as they should be over the course of development. Again, because if there's disruption of the way brain cells are communicating and connecting, that could result, again, in this inability to prune out appropriately. So as a result, you get more and more densely packed neurons, larger brain, and so you end up with a larger head size, essentially. Just a theory, but that's kind of one of the things we're working on trying to understand. Maybe that's what's happening in a portion of individuals with autism. So that brings us up to kind of where we're at today. Really, the next steps and what we're working on right now is focusing on how do we understand better these 6 genes that are accounting for about 1% of all cases with autism. How do we make sense of that so we can understand how these 6 genes are getting, how we're getting from a disrupted protein to behavior? And we have some ideas. Before I tell you those ideas, though, I'm going to just sort of recap what we've been talking about in terms of the genetics. So the current thinking is this. Even before I say that, I want to highlight one more thing. When I first got involved in this whole crazy field in the mid-90s, we knew the cause of autism in about 5% of all cases that we saw. In this time, based on all these studies, in the past 13, 14 years, we've been able to jump up to 25% of all cases of autism. So now we actually know the cause of about 25% of all cases with autism. We've come a long way. We're still far from where we need to be. We need to have answers for families. We need to make sense of this for families so we can intervene better. But we're making headway. I just want to highlight that based on the work that we've been doing. So where we stand. Structural variations, deletions or duplications, called copy number variants. These seem to be accounting for a substantial portion of cases with autism, where there's actually a structural change to the chromosome. And gene mutations seem to be accounting for another very large portion of what's happening in autism. So 2 distinct, different genetic contributions. To add to the confusion of that, these things are not mutually exclusive. So you could have a duplication on chromosome 16, and then you could have a broken gene at the DYRK1A gene. That gets complicated. And to complicate it even further, you could have just common allelic variation that's going to modify or impact the way these structural variations are expressed. When I talk about allelic variation, remember I'm talking about Gregor Mendel's P's, so a gene that codes for yellow or green, that's going to modulate or modify the impact that these structural changes or these gene mutations may have. It's just so complicated. We're getting there, but it's just so complicated. It's a question, yes?
Any of your research have any implications for why we have such an increase in autism?
That's a very good question. Excellent question. So, does this research have any implications for the sort of the reported increase in the rates? So, the first thing I'll say about that is that while the media is definitely talking about a massive rise in the rate of autism, so we need to temper that somewhat. In that there's a lot of reasons that would account for the rise in rates that we're seeing. One would be the significant changes in the way we diagnose autism. We've loosened the criteria significantly over the years. We've broadened the criteria. Second reason is the way we've done epidemiological studies over the past years has changed drastically. So back in the 80s and 90s, the way we tallied up individuals for our studies would be often through clinical referrals. And so we would tally that up. We've now, and then there was a movement towards looking at school records as a way to tally that up. And now we've sort of switched to a very effective monitoring system. So we're making headway. But anyway, so comparing rates from the 80s to rates the way we were collected 5 years ago, it's like comparing apples to oranges again. So it's hard to make that comparison. Third reason is we've got a lot more kids in the system because we have increased awareness. So we're getting kids earlier in the system and they're being recorded and tabulated in these epidemiological studies earlier. Even given that, if you account for all those things, there, above and beyond that, there does seem to be some secular increase in the rate of autism. That is, even when you account for all those things, there does seem to be some increase. Is it the 273% increase that the media is talking about? No. But from the CDC has been monitoring over the past several years using the same exact methods, and we're seeing a significant rise even within using the same methods. That's the ADDM, the Diagnostic Monitoring Network, what have you. And so we can be really confident about those data that, because we're actually comparing apples to apples in that case. And in that case, we seem to see an increase. So that's just the background. So to answer your question, though, there is some way this might play a role. So one of the things we found in our first exome sequencing paper, that sample we looked at 20 individuals and we followed up in our 200 individuals, we found that the individuals who were at increased likelihood of getting these new mutations, they were the children of older dads. So for a long time, sort of some pointing going on. Interesting in that for a long time we've known sort of the contribution of maternal age to contributing to different sort of genetic events, trisomy 21, Down syndrome, et cetera, et cetera, et cetera. We here see that these new changes, new alterations happen more frequently with increased paternal age. That's one thing, and so that we found that out in whatever 2011. We reported on that. A group in Iceland just last year actually did a study that was designed to actually look at that question and actually look at paternal age. And sure enough, they found that increased paternal age was leading to increased rates, specifically in gene mutations. And so I'm totally going to answer your questions in just a second. So that's one reason why this suggests that maybe what could be happening. Probably not count for all of it, but our changes in the way we're having children now, we're having children much later, much older, that might be increasing the rate at which we're getting these spontaneous mutations that might be contributing in some way to that risk. The other piece is, to answer your question, is we know that genes never act alone, and I'll talk about this in a moment. Genes never act alone, they always act in accordance with their environment. By environment, I mean the genetic environment, that is genes acting on other genes, but also the uterine environment. So there are, as we understand better these new mutations and these new changes that are happening, we can help sort of pinpoint then what sort of environmental contributions in utero would be impacting the expression of these genes that we know are involved. So I don't have any specific answers about that yet, but a couple years I think we'll be able to answer that question. And then there were two other questions. Yes?
What do you mean by increased paternal age? What age we are talking about?
So we don't have any good answers yet. We just see that there's essentially a correlation. The older the dad is, the greater the rate of spontaneous mutations there are. There's no good cutoff. There's no good risk factors. No good odd ratios. The way that we have a lot of data on maternal age, and as it relates to trisomy 21, we're not even there yet remotely with autism. But over 35 and up, roughly. But there's no good data on that. Sorry, I can't be more definitive. Soon. Yes? That was your question, gotcha. Yeah. Just samples aren't out there. Essentially, there's been our one study with 200 individuals, and we don't have enough people there to really make good predictions based on that. The group in Iceland also had a smaller sample size, so it's, again, not enough numbers. Soon we should have that information. Cool, all right. So, complicated story. Structural variations, as well as specific genes that are no longer functioning properly, as well as common allelic variation, all acting together in this very complicated picture, including over 800 genes, and resulting in a very complicated behavioral expression. That is the story. This is just a quick cartoon to highlight. This is just a cartoon of the chromosomes here, and each little point here with a little number and each little color signifies either a gene that's been localized and playing a role in autism, and each colored region here is a duplication or deletion at that region, leading to increased risk. The whole point of showing this slide is just to say, you can look at this, it's all over the map. There's all sorts of things going on that speaks to the genetic heterogeneity and the phenotypic heterogeneity we see in autism. Very complicated. So, that's the genetics component. as I mentioned, genes never act alone. They always act in accordance with their environment. Genes start and stop functioning at certain times based on their environment. They get environmental cues. They get cues over the course of development to say, hey gene, turn on. Hey gene, turn off. So not only are we talking about the way genes are functioning just as a static image, but also how they're functioning over time and in accordance with the uterine environment specifically. And so there's been a number of studies that actually suggest that particular exposures in utero are actually going to increase the likelihood or the risk of autism. And these are a couple that have been supported significantly in a number of studies over the years. Exposure to valproic acid, which is a mood stabilizer and an anti-seizure med, prenatal rubella, and ulcer treatment, and as well as a particular insecticide. These are all exposures that, if exposed in utero, there's an increased likelihood the child will develop autism. We do not know yet how these are interacting with any genetic predisposition, so that's the next step. The other one I just want to throw in here, just because it's gotten a lot of attention just recently, is this whole pollution idea. So this is based on work that's happening in LA. Go figure, a lot of pollution, why not study it there? Last year, a group there said, hey, let's just look at individuals who ultimately went on to develop autism. Let's look at where they were living, where their moms were living when they were during pregnancy. And they found that individuals, the closer a mom lived to very busy freeways, that there was increased likelihood that a child would develop autism. They then followed that up with another study. They got a lot of attention just maybe a month or two ago, looking at not just the proximity of freeways, but actually the amount of traffic on particular roads, thereby as a proxy for the particulate matter and pollution that would be exposed to the mom. Yes?
Does that take into account population size near major freeways?
It did, right. Good question, right. So I didn't do the study, but in sort of reviewing it, they report that they accounted for a number of factors. They accounted for SES, they accounted for population density, proximity, and et cetera. So again, I didn't do the study, but they report it in the actual article that they did count for those things. I highlight it here only because, again, it's got a lot of media attention. I think it's an interesting idea. I'm not jumping on the bandwagon yet, but I think we need to, as a field, be open to thinking about what different exposures we can have that can impact a genetic landscape. So I guess that's my point. I wish I had more to say about the environmental risk factors. We just don't know enough about it. We just don't. There's two. Yes.
Is there? I have a parent of twins who quoted a study on something I can't pronounce, but I think it starts with a T. Given during pregnancy, what was it?
Thimerosal?
No. To keep her from going into labor and to stop. So I want to call it strategy, but that's not right.
Right, gotcha, right. So I don't know that particular study you're referring to. I think what I will say that there's been a lot of one-off studies that have come out and said, hey, this is a potential contributory factor. A lot of one-offs. All of these that I'm highlighting here have been replicated at least multiple times. There are a lot of one-offs out there.
So those have all been replicated?
Yes, and the pollution one's only replicated twice by the same group. So again, I put that at the bottom there, but the rest of them are pretty, I stand behind. So yeah, there was another question. I saw a hand go up. Yes.
I wish I could remember it exactly. It was a study that came out to see the Nature Discover. It was well in the back, and you've probably seen it. It's like looking at the increase in autism reported in county by county. This was like Lincoln, Texas. It's like over a period of 10 years, and then it also showed county by county the amount of pollution, like the chemical pollution. And just to look at the county by county thing, I mean, there certainly was a correlation, whether it was positive or not, or whatever. It was very interesting to see that, and that was like, I don't know, 10 years ago. Right. So.
Yeah, I think like I said, I think we need to explore. I think we need to explore all the options. We need to be cautiously optimistic that we can figure this out. To be skeptical but open, I guess. Again, we're talking about 800 different genes that are playing a role, 1,000 different genetic contributions. Tack on 1,000 different potential environmental contributions. Lots of different things going on there. So in our last couple minutes, I want to make sure we have time for more questions. I just want to highlight then sort of how do we get from genes to behavior. The simple answer is in the brain. That's what's going on in autism. We're seeing genes that are playing a role that all have to do with the way the brain communicates. And here's just a little sort of complicated cartoon, essentially. I'll take us. I guess this isn't sophomore year biology. I don't know when you learn about the brain anymore. But anyway, we didn't learn about this when I was sophomore in biology and back at Triton Regional Public High School. But at any rate, oops. So here's a little cartoon of a brain cell or neuron. And we've got our little dendrites. This is the parts of the neuron that collect information from other neurons. On down here, the axon, which, dendrites, and then the axon here, which is going to send a message along. Here's a close-up of a synapse, the connection between two neurons. All these different little labels, genes here, these are all genes that we've identified that play a significant role in autism, primarily from exome sequencing study and this recent MIP sequencing study. And as we look through here, all these genes are playing a role in some way in the way the brain communicates. And really, that's kind of what this is all about, essentially. We identified all these genes that are playing a role. Even these structural changes contain genes that are playing a role here in the way that the brain cells communicate. The way messages are passed along, the way cells are formed, and the way they kind of move through space over the course of development as they kind of move to their place in the brain where they're going to ultimately end up. And certainly here where how they connect up and how the messages are passed back and forth between brain cells. And so that's what's going on with autism. What gets a little more complicated is that we think, well, how do we get from these specific brain connections to the complicated behaviors we see in autism? And I don't have time to talk about all the details, but I'm going to jump ahead to just a couple points about that. Well, one theory is that what we've got is, let's see, disruptions in the brain that are involved in a particular circuitry of the brain that's involved with the way we process social information. In autism, it is a disorder that I highlighted about with impairments in social communication. And so we might imagine that all the brain regions that are involved in social communication, which we've learned a lot over time through studies of primates where they actually lop off sections of the brain, which we don't do that in people. I'm glad I don't do that sort of work anymore. And actually just heard last week that NIH is actually putting an end to work on chimpanzees, actually, so that's no longer happening. At any rate, from those studies that thankfully I didn't do, we've learned a lot about the way the brain works in terms of what parts of the brain are involved in how we process information, how we utilize social information, how to respond to it. And so the theory or thinking could be is that these gene disruptions that we're seeing are impacting the way this particular social brain circuitry is functioning. And that's essentially how we get from complicated genes to the way the brain connects and communicates in particular regions of the brain that are involved in how we process social information, which is where we see the most impairments in autism. So given that, I want to make sure we have time for questions. So I'm going to jump ahead just to say quickly, all the work we've done has really been driven by all the hundreds of families that have come in and given all their time and their blood and their brains and everything to this research. And I'm always so moved every day when a family says, hey, I'm fired up, I'm really excited about participating in research because there's no way we could do any of this without parents. And so that's great. And also the sort of folks from my lab and then the collaborators as well that I work with here, it's kind of it really takes a village, is that the expression? It takes sort of a village, families and scientists working together. So on that note, I'm happy to answer any questions the rest of our time here. I want to make sure we have time for that. So thank you. Yes?
I know it's no longer from 5% to 25%, but it's still incredibly small when you look at the whole thing. So what makes you think that these patterns and trends are a cause and not just an outcome or a product or a trend that you might be seeing alongside it?
As far as the genetic findings, or?
Yeah, why would that be a cause?
That's a very fair question, very fair question. So ultimately, it is essentially a correlation. That's kind of what we're doing. I guess we're attributing cause mainly because we think about the fundamental role in the development of these structures, essentially of the person, and we think about the fundamental contribution of that of those genes to brain structure, brain function. Genes don't, like a broken gene is broken early on. And so it's not like a gene would be broken through development. It could turn on and off over the course of development, but we wouldn't see that as let's say, a broken gene by scanning DNA. We wouldn't think, oh, that's a result of something else. Because nothing in development would break a gene, essentially. Does that make sense?
Yeah, it's just that the other 75 are intact,
Mm-hmm.
right?
Mm-hmm.
So
Right. So I think what that speaks to is it speaks to there's not just one way to get to autism. So there's as I mentioned, there's broken genes, which are going to account for, again, about 20% or so, maybe a little less. These structural changes that are going to account for some percentage, and then all this common allelic variation. I still think there's something too that you get the right combination of common variation and you end up with autism. So for example, we might think about common variation being how socially motivated are you, right? So one of the predominant theories that I subscribe to about what's really happening in the brain is that in autism what we're seeing is a disruption to our reward circuitry in the brain that basically fails to assign significance to the social information, the social stimuli. If we don't assign social stimuli as emotionally valient, we're not going to attend to it. If we don't attend to it, we're not going to develop those skills. We're not going to develop the aptitude with processing social information. As a result, we may not pay attention to faces so much, et cetera, et cetera, et cetera. I got so excited about talking about that. I got to bring it back. Right, and I'm going to, as soon as I remember where I was going. So.
I get what you're saying. There's many places to look towards.
Right. Oh, right. Yes, exactly. Thank you very much for bringing it back for me. So we might imagine that there's common allelic variation that codes for how motivated you are by, or how rewarding social stimuli is to you. You could imagine someone who is incredibly rewarded and motivated by social information, and so therefore they're going to attend a lot, and that's what's going to be their focus. They're going to be totally engaged and really. But you might imagine someone else who with common allelic variation just doesn't have that same level, and so it's just common variation. It's not about a broken gene. It's just about common variation. So if you have someone who's not as attentive to common variation, they might, and again, not as pay attention to faces. They might then not develop those skills. So that's kind of my long answer to that. It's a great question, really great question. Yes?
So there are these genetic components. Do they then come and meet, so that's nature, is there a nurture piece so that they meet a certain kind of family, or has science completely ruled out nurture?
No, that's a great question. So yeah, so for me, I definitely don't see a split between nature and nurture. It's all totally connected. I think answering that question is tricky, right? For a long time, again, parents were stigmatized as causing autism. So it's tricky to sort of say, gosh, what role is nurture playing? Because there's not an ounce in my being that thinks that any way a parent is contributing to a child's autism. Do I think that people could act differently to provide scaffolding and support for a child with autism? Sure. I mean, sure. We can all sort of modify our behavior to provide a different supportive environment, but I think it's very different from playing a causal role. So, and that's actually kind of where a lot of the intervention work is going right now. So for, we know behavioral interventions seem to be effective for many children with autism, not all children with autism, but it's the most empirically supported approach is behavioral treatment following principles of ABA. There's been a strong movement away from, I shouldn't say away from, I would say toward including parents as the actual providers of that treatment. So the real focus now is not just having an individual come in for 40 hours a week and provide behavioral-based training with your child, but actually train mom and dad, hey, this is what we're doing. This is this behavioral treatment approach. You guys are the pros in your parent. You guys are the pros with how to work with your kid. These are some tips and techniques we use. We're going to train you, and you go ahead and do that. So I think there's that way that nurture can help modify the presentation. But I don't think that nurture, sort of the nurture side is playing any causal role. I think it can play a role in how the phenotype develops and how the presentation of symptoms. Does that make sense? Yeah.
What are your thoughts? Oh, sorry, I have the reps. What are your thoughts about the research about oxytocin and giving that early, and we don't know
Yeah, so certainly there's been a lot of excitement in thinking about oxytocin as a potential treatment approach for autism. Oxytocin is a peptide that is involved in a lot of different things, but one thing is it supports sort of a affiliative connection and sort of increases social motivation, so to speak, as a peptide plays that role. So a lot of folks have been excited about trying it out as a treatment approach, and there's been some preliminary work in adults to suggest that that can improve and increase social motivation and improve some of the social skills development. A lot of excitement about oxytocin and what sort of role it plays has been some involvement in the oxytocin gene there's particular allelic variants that folks are looking at, that variations of the oxytocin gene might be playing a contributory role. Jury's still out. I think we're moving forward, and I think there's a lot of promise there, but yeah, no specific answers, yeah. What's tricky about that is it's harder to get oxytocin as a treatment drug now. It's a lot easier in Europe, actually, and with adults, so that's where a lot of that work's been doing now. So, yes, you had a question, yeah.
There's a lot of talk in some circles about early nutrition and also about autoimmune correlations with AIDS media.
Yeah, so for sure. Yeah, early nutrition, there's been a number of studies that have come out recently. One was a study that came out last year looking at prenatal vitamins. So moms who aren't taking prenatal vitamins are increased risk for having kids with autism compared to moms that do take prenatal vitamins. The study was kind of a poorly done study, but it was a study that got some attention nonetheless. There was an interesting part to that study, which was that particularly there was a genetic variant. It's not something I study, but a common variation of a gene that how you process folic acid. And so it was the combination of exposure to prenatal vitamins for an individual, for a mom who had a particular allelic variant for folic acid, that those moms were the prenatal vitamins played a protective factor for the individual with this particular variant. If you had this particular variant and you didn't take prenatal vitamins, it didn't make a difference. So really it was kind of saying, if you have essentially a hit in this particular gene and you take your prenatal vitamins, that's going to play a protective role. Otherwise, if you have this particular variant, it doesn't really matter. Anyway, so I think that was interesting about that is it highlights this connection, again, sort of nature and nurture, if we want to use that term, but just the interaction between genetics and environment. So that's one aspect of nutrition. I think there's an interesting role that nutrition plays in autism in general. Certainly a lot of folks talk about the GFCF diet, the gluten-free, casein-free diet as an intervention approach for autism. And the literature out there is abysmal on that topic. The few studies that have been done are poorly done, which makes sense. It's hard to do a study where, for example, you'd want to have people blind to the outcome or blind to the intervention. It's really hard to be blind to what your child is eating. So you kind of, anyway, so there studies have been done, but in very small samples, so it's hard to extrapolate. And again, when we're talking about 1,000 different disorders, it might make sense that a particular intervention is going to work for a small number of those kids with a particular genetic background, but if you're looking at 1,000 different kids and only work for 4 or 5 of them in a big study, we're not going to find out. The point is PKU, again, is a great example of there was a dietary intervention that completely alleviates the behavioral presentation, the phenotype, but only in a very small portion. So again, if we take 1,000 kids with autism and let's say prior to us understanding what PKU was, that's going to account for 0.5% of kids with autism. You do a study where you take away phenylalanine from all 1,000 kids with autism, you're not even going to notice that you're actually that you changed the phenotype in the small 0.5%, and that could be what's happening with our current diet intervention studies, is we're not mapping those onto the appropriate kids. There's a lot of sort of things I'm throwing at you about regarding nutrition and information here. So I think we just don't know enough yet. I mean, the take-home message, I think there's a lot we need to do to make sense of that better. So that's your question about nutrition, but there was a second part of that question as well. Autoimmune, right, exactly. So we do know that, that there's increased rates of autoimmune disorders in family history of individuals with autism. We don't know much more than that yet, actually. There's a lot of theories out there. an interesting op-ed in the New York Times about 2 months or so ago that was definitely an editorial, but was written with great authority. And I actually got a little concerned when I read it because it was really based on this a well-written theory, but not a lot of science behind it. So I think there's some great theories about thinking how what role autoimmune disorders might be playing a role. We just don't know yet exactly the mechanism. So I wish I had more answers for you than that. To say that I think we need to explore both those issues more closely. But that's as much as I can say. Sorry. Any other questions? Yeah. Oh, sure. Yeah, thank you.